Welcome everybody to Bobo's Biology channel, where we help you become an academic weapon just before your exam. On today's episode, we discuss glycolysis. Before we jump into today's topic, let me tell you a little story. You see, Bobo the curious mouse was running on his wheel one day, feeling full of energy, when suddenly he wondered, where does all this energy come from? He had just eaten a delicious piece of cheese. But how did that turn into energy that he needed to run and explore? That's when he started thinking about glycolysis. Glycolysis happens in the cytoplasm whenever a cell needs energy, especially when blood glucose levels are high. It's a continuous process occurring in most cells to ensure they have enough energy to function. Since glycolysis doesn't require oxygen, it can take place in both aerobic and anaerobic conditions, making it a fundamental energy-producing pathway for all living organisms. For example, when Bobo's muscle cells need energy fast, glycolysis kicks in immediately to break down glucose and produce ATP. ATP is then used to allow Bobo, the curious mouse, to run on his exercise wheel. Energy is released when a molecule moves from a high energy state to a low energy state. The bonds in glucose store a lot of energy, but this energy must be harnessed in a controlled way. Glycolysis is a 10-step process that gradually breaks down one glucose molecule, which is composed of 6 carbons, into 2 pyruvate molecules, which are composed of 3 carbons each. But why is this breakdown controlled? And why split glucose into 2 3-carbon molecules instead of 6 1-carbon molecules? When glucose is burned, it rapidly breaks into single carbon units, releasing energy as heat in an uncontrollable way, wasting energy and generating temperatures too high for the body to handle. Glycolysis, in contrast, allows cells to extract energy efficiently in small, usable steps in the form of ATP or NADH. Our journey begins as a glucose molecule, a member of the aldose sugars, marked by an aldehyde group on its C1 arm. The molecule is found in its cyclic structure. For our purposes, we will show here both the Fischer and the cyclic structures for the molecules that can form both. Glucose must first enter the cytoplasm of the cell. The blood transporter carries the glucose via facilitated diffusion, a type of passive transport, into the cell's cytoplasm. After glucose is transported into the cell, the glucose requires a tag in order to avoid getting spit out of the cell via the same blood transporter, because it's a passive transporter. This tag is a phosphate, which is added by hexokinase or glucokinase in the liver cells on the 6th carbon of glucose. The phosphate comes from ATP, which is converted into ADP. This process forms glucose 6-phosphate, as can be seen here in the Fischer projection on the right, and on the left in its cyclic structure. This is the first phosphorylation step, meaning a phosphate group has been added to the molecule. This could be also considered as the first energy investment. In the next step, glucose 6-phosphate will be isomerized by glucose phosphate isomerase into fructose 6-phosphate, making a ketose sugar. A ketose sugar, meaning a sugar with a carbonyl group in the middle of the carbon chain, in contrast to the aldose sugar, where the carbonyl group lies on the end of the molecule. The phosphate group remains on carbon 6 during this process. This step is called isomerization of glucose 6-phosphate to make fructose 6-phosphate. In the next step, fructose 6-phosphate is phosphorylated by phosphofructokinase, also called PFK1, taking the phosphate from another ATP, which turns into ADP. This leads to the formation of fructose 1,6-bisphosphate. The bis in bisphosphate means that the 2 phosphate groups are not directly next to each other. The new phosphate is added on the C1 carbon, as can be seen here in the Fischer projection on the right and on the left in its cyclic structure. This step is the second phosphorylation and can also be considered the second energy investment. In this step, fructose 1,6-bisphosphate is cleaved by aldolase into two 3-carbon molecules named glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The phosphate on glyceraldehyde 3-phosphate is on the third carbon, as can be seen here, and the phosphate on dihydroxyacetone phosphate can be found on the first carbon. These two molecules and all following molecules cannot be presented in cyclic structure anymore. This step is called the cleavage of fructose 1,6-bisphosphate. Dihydroxyacetone phosphate in the process of glycolysis has to be converted into glyceraldehyde 3-phosphate to be further used in the process. This is facilitated by triose phosphate isomerase, which marks the isomerization of dihydroxyacetone phosphate. Therefore, two glyceraldehyde 3-phosphate molecules will partake in the next steps. This step is called the isomerization of dihydroxyacetone phosphate. The next step involves the conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase. In this process, one inorganic phosphate is being used and one NADH is produced. This process occurs twice since dihydroxyacetone phosphate from the previous step was turned into glyceraldehyde 3-phosphate. So all following steps will happen once for each molecule of glyceraldehyde 3-phosphate. Therefore, two 1,3-bisphosphoglycerate and two NADHs are being produced and two inorganic phosphates are being used. This step is called the oxidation of glyceraldehyde 3-phosphate. This step is where the energy investments of ATP in the beginning start to pay off, where 1-bisphosphoglycerate is dephosphorylated by the enzyme phosphoglycerate kinase to produce 1 ATP and 3-phosphoglycerate. This step occurs twice, since two 1,3-bisphosphoglycerate molecules are starting into the reaction. The phosphate that was lost in this step was originally at the C1 end of the molecule. Therefore, this step can be classified as a dephosphorylation reaction. In the next step, 3-phosphoglycerate is isomerized into 2-phosphoglycerate. The phosphate is thereby just transferred from the C3 carbon to the C2 carbon. This reaction again occurs twice, and this step can be classified as an isomerization reaction. In this step, the molecule 2-phosphoglycerate is being dehydrated by enolase into phosphoenol pyruvate. This step occurs twice, and two molecules of H2O or water are being lost. Thereby, the step can be classified as a dehydration reaction. In the last step of glycolysis, phosphoenol pyruvate is being deprotonated by pyruvate kinase into pyruvate by producing 1 ATP. This step again occurs twice. Thereby, in this step, 2 ATPs are being produced. This means that from a single glucose molecule at the start, we have produced 2 pyruvate molecules along with 4 ATP molecules in total. However, we used 2 of those ATP molecules in the initial investment stages, and therefore the net gain of glycolysis is 2 ATP and 2 NADHs. Under aerobic conditions, meaning where O2 oxygen is present, pyruvate can enter the mitochondria and can be converted by the pyruvate dehydrogenase complex into acetyl-CoA. And acetyl-CoA can be incorporated into the Krebs cycle, or also called Calvin cycle, which will ultimately produce NADH and FADH, which will lead to the production of ATP in the electron transport chain. Under anaerobic conditions, pyruvate can be acted on by lactate dehydrogenase, which utilizes two hydrides from two NADHs, forming lactic acid, which in the heart and brain can be utilized as a direct energy source. Remember Bobo running on his wheel? When he's sprinting and his muscles outpaces his oxygen supply, glycolysis still provides energy through anaerobic respiration, producing lactic acid in the process. The lactic acid produces that burning feeling in his legs when Bobo is running on his wheel. Since in muscle cells, lactic acid cannot be used as a source of energy, once oxygen is available again, lactic acid will be transported by the blood to the liver, which then undergoes the Cori cycle and produces glucose again. This glucose molecule is then available for glycolysis again. If there is plenty of oxygen, the process continues into aerobic respiration, leading to the production of more ATP in the mitochondria. Overall, glycolysis occurs when a cell needs energy, especially when glucose is available in big portions and needs to be broken down for ATP production. It's the first step in cellular respiration and happens constantly in most cells to ensure that they have enough energy to function. Let's quickly recap what we said so far. So, in the first step, we transported glucose into the cell and added a tag in the form of a phosphate from ATP, forming glucose 6-phosphate. Then, in the second step, glucose 6-phosphate was isomerized into fructose 6-phosphate. In step 3, fructose 6-phosphate was phosphorylated by another ATP, forming fructose 1,6-bisphosphate. Then, in step 4, an aldol cleavage splits fructose 1,6-bisphosphate into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. In step 5, dihydroxyacetone phosphate had to be isomerized into glyceraldehyde 3-phosphate to be further used in the glycolysis process. In step 6, two molecules of glyceraldehyde 3-phosphate are being oxidized into 1,3-bisphosphoglycerate, while two NADH molecules are being produced. In step 7, two molecules of 1,3-bisphosphoglycerate are being dephosphorylated into two molecules of 3-phosphoglycerate while producing two ATPs. In step 8, two molecules of 3-phosphoglycerate are isomerized into 2-phosphoglycerate. Then, in step 9, two molecules of 2-phosphoglycerate are being dehydrated to form phosphoenopyruvate. In step 10, phosphoenopyruvate is dephosphorylated, forming two pyruvate molecules and producing two ATP molecules. Under aerobic conditions, pyruvate can be converted into acetyl-CoA, which can then undergo the Krebs cycle and ultimately produce ATP during the electron transport chain. Under anaerobic conditions, the two molecules of NADH produced during the glycolysis reaction will drop off their hydrides onto pyruvate again to form lactic acid, which will be utilized depending on the type of tissue. And that's glycolysis, turning sugar into energy so you don't have to. If you like this video, hit that like button. Think of it as your way of making ATP for this channel. And subscribe, because next time we're talking about how glycolysis is actually regulated. Turns out your enzymes aren't just eyeballing it. See you then, and stay curious.